All right, Ren, are you ready?
Yep.
Wonderful. All right, for everybody online, thank you so much for joining us today for HMSC's research seminar. My name is Cinnamon Moffett, and I'm the research program manager at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon, and I'll be your host for today. Just a couple logistics before we get started. Wanted to let everybody know we are on a Zoom platform, but we do have your mics, camera, and screen share disabled to help folks that have limited bandwidth. But we would love for you to chat with us using the chat box function, which you can find at the bottom or the top of your screen. And today's speaker would really like you to put in questions whenever you have them, and we will answer them as we go along. So feel free, if you see something, go ahead and put that question in, and we'll answer it right when that topic is being talked about. So feel free to use that chat. I also wanted to let folks know that we are recording today's seminar, and it'll be up on the HMSC's past seminar page in a few days. And I am putting in that link in the chat box right now. So if you miss something or you would like to share this or pass it along, you're welcome to go in and find a recording of today's presentation at that link. So the other thing I wanted to do was just do a little promo for next week, same place, same time, just next week. On May 6th, we have Beth Phillips from NOAA's Northwest Fisheries Science Center at NOAA, which I already said, that's going to be talking about using archival data to expand the understanding of the California current ecosystem. So hoping that you can come and join us again for that talk. But if you're interested in any of our Hatfield events, you can go to Hatfield using Google or whatever platform you use. Scroll to the bottom of our landing page, and there's a calendar of events with all the login details that you might need to join us for one of those events. I also wanted to just put a little shameless plug out that I am now booking for summer seminar. We're going to keep this virtual format through the summer. So if you or one of your colleagues are interested in giving a talk, please reach out to me, and we'll see if we can get you on the seminar schedule. We're probably going to go to a hybrid format in the fall. So we'll see how we keep going, but if you'd like to join us for summer, please reach out to me. I would love to see us continue to have some wonderful speakers like we have today. So that's what happened with Aaron Galloway. He's actually the individual that suggested that we have today's speaker. So thank you, Aaron. I saw you on the line. But for today, I just want to introduce, since you're all here for this, this afternoon we have Reyn Yoshioka, who is from OIMB at the University of Oregon, who will be talking about dock shrimp. But first, just a real short little background about Reyn. Reyn grew up in Hilo, Hawaii, and started his research career with Dr. Drew Harville as an undergraduate and lab tech at Cornell University, working on projects such as studying coral and sea star and eelgrass diseases. Reyn is now a fifth-year PhD student with the Coastal Trophic Ecology Lab of Dr. Aaron Galloway, at the Oregon Institute of Marine Biology, and he's going to be sharing his work with us today. So Reyn, the floor is yours. Take it away.
Awesome. Thanks, Cinnamon. Well, thank you all for having me. I'm going to be talking about some of my dissertation work looking at a rhizocephalan parasite of shrimp. So without further ado, there we go. Okay. So what I'm going to cover today is, of course, just introducing you to the cast of the projects that I'm working on. So the Rhizocephala more broadly, Pandalus, which is the host of the parasite system that I work in, and then Cylon, the parasite. I'll cover two different projects with it. First is the host with the most, and that's looking at epibiosis and Cylon parasitism. Then going into You Are What Eats You, and talk about the fatty acids of Cylon-infected shrimp. And then lastly, I'll just wrap up, and there'll be more time for questions, or as Cinnamon mentioned, please do add your questions in throughout the talk, and we'll try and answer as soon as they come. So diseases and parasites and pathogens by definition are bad. They have negative effects on their host. And of course, that's the same kind of visual that comes to our mind, right? These things are bad things in the environment. But it's also important to remember that they're parts of their ecosystems, right? And they can have cascading effects on other ecosystem members. And so, for example, here, this is probably one of the most on our mind cases for disease in marine ecosystems. This is a ochre star that has succumbed to sea star wasting disease and has basically melted into a blob. But whether it's affecting this particular species as a keystone predator or other predators that exert really important roles on their ecosystems like Pycnopodia, it's really clear that diseases, parasites, and pathogens can have important roles in their ecosystems. But it's not always this totally destructive thing. There are parasites all over the place. They're the most common consumer strategy in biology. And so it's important to think of them also just as other ecosystem or community members. And that's how I tend to think about them in my dissertation work. So the way that I do this is that I frame the host in the context of its food webs, placing it in its relationships with its predators and with its prey. But of course, we know that the hosts aren't just the host, right? There is some portion that is the host proper, and then there are all the symbionts that go along with it. And this includes the parasites. And particularly in the case of parasites, there's an extractive relationship where parasites benefit at the expense of the host. And in other symbiotic relationships, of course, there can be some benefit to the host as well, but there's always this give and take. But their relationship sits in the context of the host's relationships with other organisms. And so I'm curious to figure out what parasites do to their hosts and how it affects their host relationships, especially with the relationships of the predator, whether it's the strength of the relationship, so how much a predator might be eating this host along with its parasites, but also the flavor, whether that relationship shifts a little bit or if a infected host means something different for a predator. So the system that I study uses barnacles as the parasite. So this is a typical, acorn barnacle, giant acorn barnacle with a bonus nudibranch. We have gooseneck barnacles of both the anchored on the rocks type and then the pelagic floating or on-floating items flavor. But these aren't the kinds of barnacles that I'm actually going to be talking about. I'm talking about these ones here, not the hermit crabs or the shrimp that are shown, but those little blobby things that are attached to them. And so those blobby things are actually the external parts of a barnacle parasite that infects other crustaceans. And so this group called the Rhizocephala they're really well known for being these spectacular barnacle parasites of other crustaceans. And so this here is a Leuckart wall chart made by Rudolf Leuckart, who is known as the father of modern parasitology. And it shows a great example of what we tend to think rhizocephalans look like. So the big thing that it shows is that adult rhizocephalans are pretty darn morphologically reduced. And there's two main body sections that we're going to be concerned with. The first is most apparent to us, and that's because it's on the outside, appropriately called the externa. And the externa is where all the reproductive fun happens. And so the young are brooded in the externa and eventually get released to go on and infect other crabs or, in my case, shrimp. Now, the other body section that the rhizocephalan has is the interna. And this is a root-like structure that penetrates the host and draws nutrition and redirects it towards the externa. And so rhizocephalans are known as parasitic castrators or sterilizers because by taking their host nutrients, they redirect the reproductive investment that a host would have towards making its own young and shunt it off to reproducing parasite babies. And so it's a really spectacular relationship. And the reason that they're called rhizocephalans is because of this kind of branching root-like structure that the interna takes into the body. So rhizocephala meaning root head. And so in redirecting the host's resources to the parasite's young, it does a really neat thing called brood mimicry. And so on the left side, you can see a crab that's infected with a rhizocephalan, and the externa sits right in the same location that the crab's eggs would be, and that's shown on the right panel, so A. And it's not just these reproductive structures taking the place of the eggs for the host. But what will actually happen is that the host behaviors will shift and the host will start caring for the externa of the parasite as if it were its own young. It'll ventilate it and groom it. And even more so, it's not just the female crabs or the female hosts that this happens to. The male hosts will sometimes get feminized, and that means that they'll start to take on both those behaviors of caring for the externa or the fake eggs, but sometimes their appendages will start to develop female features, and in some cases the abdomens will broaden, and so they'll look more like a female crab. And so rhizocephalans have these really spectacular effects on their hosts, and so that just makes them really interesting in terms of just thinking about how wild biology is. But with that, kind of wondered, like, are these really barnacles, right? They are morphologically reduced. They don't filter feed, they're parasites. But hopefully I can convince you really briefly that they are. So this is a tree of the Thecostraca, and the branch that we're concerned with are the Rhizocephala here. And the Rhizocephala sit within the clade Cirripedia, and both of those other branches of the clade, including Thoracica, which are typical barnacles, and Acrothoracica are filter feeding. And this suggests that Rhizocephalans in belonging to this clade evolved from a filter feeding ancestor. And so it's kind of an evolutionary feat that they've become so well adapted to parasitizing other crustaceans. So setting the place for where this work was done, this was up at Friday Harbor Labs in Washington. And I was actually up there to do other work. And this started off as kind of an odd side project that I was lucky to be able to do while we're actually up there doing other work. And eventually this culminated into becoming part of my dissertation work. So if there are any students on here, hopefully I don't anger your advisors, but keep your eyes peeled for neat things that are happening right below your feet. You never know what kind of neat biology you'll discover. So if you go up to Friday Harbor Labs and you look off the docks, you'll see this familiar critter over here. This is the dock shrimp Panelistani, and there are just heaps of them hanging out under the docks. What's even more remarkable is that there are also heaps of them that are infected with their parasite, which is cylon hippolytes. So cylon hippolytes usually makes one externa, but you see here that there are two bulges popping out from underneath the shrimp, and so it's a bonus. And this represents probably two different females that have infected this one shrimp host. So for me, I was really interested in these because one, my research up until this point has been concerned with diseases and parasites, but in joining the CETA lab, the Coastal Trophic Ecology lab with Dr. Aaron Galloway and my PI, I wanted to look at those relationships in the context of food webs. And so we use fatty acid analyses to study those food web relationships. And here there was a abundant host with abundant parasites that I could easily go and collect and analyze for fatty acids, which we use for our work. And so I went off and started collecting them. And I'll get to those data in a bit, but something else more interesting came up. And so this brings us to part one, which is talking about the host with the most. So as I was collecting these shrimp, I was just starting to look at them and appreciate how beautiful they were just simply as animals, because what's the point in doing science on an organism if you can't experience it? And what you see here are two dock shrimp. The one on the right looks what we imagine dock shrimp tend to look like, and it has a very smooth carapace. The colors are relatively bright, it's light colored, whereas pretty much everything that I just said is not true for that poor shrimp on the left. It, for lack of a better word, looks scuzzy. And so what I was seeing were things like this, like plumes of algae that were growing on top of the shrimp's rostrums. In other cases, there were sporid worms or sometimes even bryozoans growing on top of these shrimp. Even sometimes those typical barnacles, which I thought was really, really neat. And then if we look closer, this is a microscope image of some of those algae growing on top of the rostrum of a shrimp, or this stringy brown alga that was growing on top of the antennae. And then this here is Ephelota, which is a ciliate that is actually the most common falling organism or epibiont that I saw on the shrimp. And in this case, it's growing on top of the shrimp's tail. And so in finding these epibionts growing on these shrimp and just kind of wondering like what was going on with them, I started to notice another thing, and it was that the shrimp that had epibionts were the shrimp that also were parasitized and had really clearly that cylon externy on them. And I hadn't collected very many shrimp up until that point, and so I wasn't sure if there was something actually going on here. Was there truly an association between the cylon parasitism and epibiosis of the shrimp? And so just went and did the old-fashioned collect a lot of shrimp and measure them strategy to get to the bottom of this. So what I did is I collected 575 shrimp off the Friday Harbor Labs docks, and I did this with a specialized net, which had a flexible opening that I could run up against the side of the dock pilings and slowly bring it up and collect the shrimp along the way without scaring them. And this helped me get a relatively unbiased capture of all the shrimp that I then measured for the presence and absence of different kinds of epibionts, the size of the shrimp, the sex of the shrimp, and then also the degree of cylonization or the stage of the cylon on the shrimp. And so by collecting all of these data, I was then able to fit a set of logistic regressions to try and understand the relationship between cylon and the epibiosis. And so that's what I'll be showing here. We'll first start off with Ephelota, that ciliate. That was the most common epibiont that I saw. And on the x-axis, we'll put the stage of cylon infection. One of the nice things is that with these shrimp, their undersides are clear enough that you can actually see what's going on. In other situations with rhizocephalans, it gets a little bit harder because you might not be able to see what's going on internally for the rhizocephalan, and you just have to rely on the externa to try and gauge parasitism status unless you dissect the shrimp. And so luckily I was able to do this without sacrificing all 575 of my shrimp. And so if we go along, we can see this is the underside of one of the shrimp. An uninfected shrimp has pretty clear looking tissue. There's not a lot going on. It's glassy. It looks normal. When the cylon starts to infect the shrimp, so after the shrimp gets infected, it starts to develop and you see these web root-like structures starting to form, and it gets a little milky. Eventually, that forms a primordial externa. So that's before that external part actually emerges from the shrimp. It's still underneath the cuticle, and you can kind of just see a little bit, or a little dot. And then after the shrimp molts and the externa develops further, it'll actually come out of the shrimp and you'll have the externa proper. In this case, you get, again, a bonus because I had seen so many single externae on these shrimp that I didn't think to take a picture of those on the underside for the series. But this case, there were two of them, and that was neat. And so you have a picture of two externae on a single shrimp. But after that externa develops and releases the larvae, it kind of shrivels and wastes away and disappears, and the shrimp is left with a scar. And so by staging the shrimp, or staging the cylon for all the shrimp this way, we can get a proxy for the duration that a shrimp has been affected by this parasite. And so that's why we have it on the x-axis. So progressing from left to right. So on the y-axis, we'll put the predicted probability out of these logistic regressions of having the Ephelota as a falling organism. And because I collected presence and absence data, we can actually put those on top, where presence are all of those ticks at 1 and absence are all of those ticks at 0. And then finally, after fitting the model, we can put our predictions. And so those are the points there. The error bars are the standard errors for those predictions. And I sort of expected it, but I didn't expect it to be this straightforward, is that there was very clearly an increase in the probability of Ephelota being present as a falling organism on these shrimp. what causes the ephyllota to disappear? So if I interpret this question correctly, it's, or actually, John, would you mind clarifying? Disappear in what sense? To leave the shrimp?
All right, Reyn, go ahead and continue, and we'll either let John add in his comments or we can unmute him. Oh, there it is.
Oh, gotcha. Okay. So the externa. So after the externa, well, the externa disappears when the larvae have been released. When there's a scar, that scar can, well, we're actually not sure if cylon persists as an infection then, or if it's a one-time thing where it just makes an externa and then it disappears. I have seen one case where a shrimp has molted after having a scar and there's been no signs of cylon infecting the shrimp. But then in other cases, I've seen those shrimp being a lot more, in a way, fragile. And those are the ones that didn't particularly survive the survey, or in the holding tanks that I had the shrimp in, were the ones that got attacked by other shrimp and then were cannibalized. So I'm actually not sure whether the cylon infection is persistent, but the externa does go away and the scar can go away. Hopefully that answers your question. Okay. So it was nice to see, or it was interesting to see that there was this really clear increase. And this persisted also with the other following organisms that I saw, or that were quantifiable. There were some small red algae, some lacy red algae, and some flat red algae. And in all those cases, the later, or the longer the cylon had been infecting the shrimp, the greater the probability of that shrimp having these different following organisms. One thing to point out, and I'm sure some of you might have noticed, is that I've just given these really simple morphological names. So they're just really just categories. And that was because one, I don't have enough expertise to identify little tiny red algae as cool as I find them, but also there are 575 shrimp. So that would have been a little invasive, and I really didn't want to kill as many shrimp as I could have had I done all of this. Okay, so the shrimp were left because of their parasitism as being fabulous. They had a lot of bling. Is that outdated? Probably. But they had the algae, they had the ephyllota, and this was really neat, just seeing how much a rhizocephalan could affect its host. But that leads us to ask, why is that the case, right? So the most straightforward explanation is that the molting of the shrimp was disrupted by having a rhizocephalan, or the cylon infection. And this is a common occurrence with a lot of the other hosts of rhizocephalans, where their molting will be completely seized, or the intermolt period will be extended. And so that means that without the host molting and basically casting off the epibionts that had grown on it along with the old molt, there's a longer period of time for those epibionts to settle onto the host and accumulate. And this is hypothesized to be the case with increase in epibiosis rates for crabs. Other things that happen is, or could be behaviors that get changed because of cylon infection. And so in other rhizocephalans, again with crabs, a lot of the research has been on crabs. As I mentioned before, of course, there are the brood mimic behaviors that the crabs take on, but also in some situations, crabs will burrow into the sand less. And so by burrowing, a crab kind of, in burrowing, the sand scours off the epibionts that were living on top of the crab. And so without having the normal burrowing behavior, epibionts can again accumulate onto the crab. And so that's another reason that these rhizocephalans could be affecting epibiosis through the behavior. In my case, I'm really curious in trying to understand whether the grooming behavior has been shifted by cylon, whether there's some trade-off in activity that the shrimp has to dedicate to the cylon, either just by staying still to reserve energy or whatnot, or actually changes in the shrimp's grooming behavior, because shrimps, and especially Pandosani, have evolved some sophisticated activities for grooming different parts of their body with different parts or with different appendages. And so disruptions in those grooming activities could be really important in fending off epibionts, and that might be shifted because of rhizocephalan infection. But a lot of speculation, but hopefully not speculation soon. So we're hopefully getting to the answer for that. Sun Brown is working with us, analyzing videos that we have of shrimp that are and are not infected with cylon, and they'll be comparing the activities and the different grooming activities between the infected and uninfected shrimp. So hopefully we'll know if behavior is getting shifted because of rhizocephalan infection. And then as I mentioned before, I don't have personally the expertise to identify these small red algae, but many of you may know Nancy Treneman, who is a wonderful naturalist and fortunately does have the expertise. So she is working with us to identify the algae that I was able to sample. And so I'm really glad to have these folks joining our team on this project. So that's all fine and dandy. Like, it's really neat to think about how a rhizocephalan is changing its host as kind of a raft for other organisms. But as I mentioned before, I'm really interested in the relationship that these hosts have with their predators. And so there's some reasons to believe that through epibiosis, cylon might be indirectly influencing predation rates on top of these shrimp. And so first off, the reason that these shrimps have evolved grooming behaviors is hypothesized to be that they need to be able to swim well. So both for just general locomotion, but of course for getting away from predation, these shrimp need to be hydrodynamically efficient so that they can get away. And adding these epibionts could increase drag or just general weight to the shrimp and inhibit that. Another interesting thing could be just direct interference with sensing. So this is an example of one of the shrimp that had just its eyes completely covered with falling organisms. I presume these to be diatoms. But it wasn't just the eyes. Sometimes it were the antennae that were covered in epibionts. And so both of those are important sensory organs and would basically warn the shrimp from its predators. And so if those are interfered with, then that could be another reason that fish might get more of a shrimp snack. And so that kind of leads us into the next topic, right? Trying to understand this relationship. And the way that our lab does this is with fatty acid analyses. And so in this case, our mantra, or in a lot of cases, our mantra when we deal with fatty acids are you are what you eat. But in this case, it's you are what eats you. And so I was just so excited to find these shrimp because I could try to get to this consumer resource relationship through the context of this parasite and its host. All of the resources that this parasite is getting are coming from a single host. And so there's a one-to-one direct trophic relationship that's happening here. That isn't common with a lot of other systems that we study. And they tend to be really complex, right? Food webs have a lot going on. A lot of different organisms are eating a lot of different organisms. And so that presents a complication when we're trying to understand food webs, especially when we can't directly observe the activities of a given organism or species. And so the way that our lab and many other labs do this are using biomarker approaches, in this case, fatty acids. And so what this shows here is a diagram of kind of how that might happen. Different primary producers produce particular kinds of fatty acids, and those are the different colored circles or pie pieces in the diagram. And when an organism consumes those materials, depending on those fatty acids, they'll likely be retained. And then you can follow those different fatty acids through a food web to try and understand trophic relationships that are going on. And so this is a really useful approach when we're trying to understand food webs. But one of the difficult things is that in most situations, a given organism is consuming a mixture of other organisms. And so it takes a lot of validation to try and make sure that that conversion of the fatty acids in the prey are also reflected in the predator. And that's why this shrimp was so exciting to me, is because it was that one-to-one relationship where we don't have a complication with mixing. But another thing that fatty acids also tells us in the system is about the lipid dynamics that are happening because of the parasite. What is the parasite taking away from the shrimp? How is it redirecting lipid and fatty acid resources towards its own benefit? And so I was hoping to try and get to the bottom of that by collecting some of these shrimp for fatty acids. And so this happened in kind of two sets. The first sets are whole shrimp that were either parasitized with really well-developed externi, and then other ones that showed no signs of having rhizocephalans. So I even looked underneath them and only chose the ones that didn't have any of that kind of wispy root-like interna that would have cued me off to their infection. In another set, I collected 10 each of infected and uninfected shrimp and dissected them. So I collected the hepatopancreas, which is basically the shrimp's liver. And that's where a lot of lipid metabolism and storage happens. And so that tells us what the shrimp's lipid energetics are kind of doing. And then I also collected the abdomen, which also has some of the rhizocephalans interna penetrating it. And so we have a mix of both host and parasite tissues then. And then the easiest source of purely parasite tissue was the externa. And so we're also getting a picture of the lipids that go into the rhizocephalans reproduction, as well as its just general lipid biology. And so then after collecting those fatty acids and extracting them and getting the data, what we end up with in our fatty acid analyses are dozens of different fatty acids. And you can imagine that trying to analyze each of them separately is quite a daunting task, and it'd take up way more of the screen than I could ever show. And so one of the strategies that we do to deal with all these different kinds of fatty acids is to use multivariate analyses and ordinations so that we can better interpret what's going on with the collective sets of these fatty acids for these different samples. And so that's what I'm showing here. This is a non-metric multidimensional scaling plot, or NMDS. And so basically what I'm doing is I've taken all of those different fatty acids, all of those different dimensions, and compressed them into a two-dimensional space that's easier for us to interpret. And so what you'll see here are different points, and each of those points represents a single sample. The different shapes represent different tissue types. If it's a closed circle, it's whole shrimp, and then the rest of the symbols are the different dissected tissues. And then the colors are the cylon status. So either blue is cylonized or is infected, and then the kind of minty green are the uninfected shrimp. And so that's here. So those arrows are vectors showing the relative correlations of different particular fatty acids that were shown to be important in these analyses, their correlations with the NMDS dimensions. And so you can think about different points going off in those different directions, showing more contributions of that particular fatty acid to those samples. The other thing to remember when you're interpreting these is that the closer points are together, the more similar they are in their suite or profile of fatty acids, and the farther they are apart, the more different they are. And so the first thing that I want you to take away from this is first that the cylon externi are pretty distinctive, and they move away from the rest of the samples in the study. One that's probably because they represent a different species, right? Because these are just parasite tissues. And then the second thing is that they are also reproductive tissues. And so you might expect them to have pretty distinct fatty acid profiles relative to the rest of the samples. And they move off in the direction of oleic acid, which is a really common and abundant monounsaturated fatty acid for those of you that are interested. The second thing that I want you to see is that the whole shrimp are distinct in their fatty acids. So the cylonized shrimp and the uninfected shrimp are distinct from each other. And this, for one, was pretty interesting because it showed that a parasite could affect its host fatty acids. And the reason for that is probably because of this contribution of the externi here, because they move off in the same direction, the infected shrimp, I mean. So as I keep asking, does this really mean anything? Are these fatty acid changes meaningful? And it's hard, especially in this space, to try and get at those questions because we have pretty slim ideas of what these fatty acids are relative to another organism or something like that. And so one way that we can get to answering whether these actually mean anything for us or the other organisms that live in these communities is by doing some hypothetical analyses, for lack of the actual empirical work. So what I'm doing is a hypothetical diet analysis of copper rockfish, and I'm using the quantitative fatty acid signature analysis method, or QFASA, to do this. And so what QFASA does is it's basically a mixing model that can help us understand the contributions or the proportions of different prey to the diets of a predator, given the predator fatty acids and given the prey fatty acids. And we actually analyze a suite of those different fatty acids to try and get to the bottom of this. And so on the x-axis, there's different prey that I use. So there's sand lance, herring, rock crab, Dungeness crab, and then shrimp. So the shrimp are from this study, but the rest of the prey, as well as the copper rockfish, those fatty acids are from literature sources. So this is very much hypothetical, but hopefully will tell us something about the system generally. And so on the y-axis, we get the predicted proportion of rockfish diet based on QFASA. And if we run it, we can get our predictions along with their standard errors there. We see that herring contributes the most to the rockfish diet, at least given the data that we have. And then that cylonized shrimp are actually the next most abundant prey type in this diet. You also see that the uninfected shrimp have a relatively low contribution, but that's almost expected because we know that they are still pretty similar in terms of their fatty acids, despite being distinct. And so when you run analyses like this, as you get more of one, you almost have to have less of the other. And this kind of shows up if we run it and lump all the shrimp together, where we see that the shrimp that are predicted here are more or less the addition of the uninfected and the cylonized shrimp. So that kind of makes sense. All the other prey types are pretty similar. So that's not super informative as telling us what parasitism means in this system. And so I really wanted to try and manipulate, especially since this is a hypothetical situation, manipulate the amount of cylonized versus uninfected shrimp and see how the QFASA model changed. And so the way that I did this is I took those 12 shrimp that I have data for that are cylonized and the 12 shrimp that I have that are uninfected and made different combinations of them to get a set of ratios of cylonized to uninfected shrimp. So just generating a bunch of different data sets. And so for example, in this case, we'll look at 9 to 3, cylonized to 3 uninfected, and then I could subsample those shrimp to get a set of the 9 shrimp that I'm going to use in this particular data set, and then also subset another 3 from the uninfected so that I have the correct ratio. And ultimately, the sample size still adds up to 12. But because just this one random sample could have just pulled for no good reason some extreme values, I didn't want to just rely on one subsample each. So I did it again. And then I did it another 98 times so that for this 9:3 ratio, I have 100 iterations, 100 subsamples that I could run through QFASTA and get predictions out of. And then I did the same thing, so all of those 100 times for all the different ratios of cylonized to uninfected shrimp. So 12 to 0, all the way to 0 to 12. And then so I could feed those to QFASTA and see what the model predictions look like. And so within each of those different prey types, the X and Y axes are the same as before. We get 13 different dots, and each of those dots are one of those combinations that go from 0 to 12 to 12 to 0, cylonized to uninfected shrimp. And so as you move from left to right, you get an increasing number of cylonized shrimp. So, and it was really neat. I actually was hoping for this, but I totally was not expecting that something would actually happen. There seemed to be a directional shift based on the changing the fatty acids, or the proportion of the shrimp that were parasitized in those fatty acid data sets. And some of the shifts were relatively large. So that was an 8% shift in the herring, their contribution to the rockfish diet. Even though if you look at the shrimp predictions, they go up and then they go down, but more or less they're the same. And so one thing that cues us in is that not taking into account the parasites in the system might not change the predictions for that particular species that we're most interested in this case, the shrimp, but it may change the predictions for everything else. And this plays into how we try to understand food webs from a biomarker perspective more generally. Are we taking into account the parasitized individuals? Are we taking into account the different prey that may not be what we think of as the most typical or normal looking prey? And so to better explain that, say I had actually done this analysis and collected all of the different prey types as well as the rockfish. Well, I would have collected, pretty much normal looking rockfish. And for all the different prey types, I would have collected, the most typical looking herring, the most typical looking sand lance. And in the cases of the shrimp, I would have chosen the most typical looking shrimp, which to me wouldn't have been the ones that had parasites. And as this shows, our predictions change if we don't account for the parasites. And especially if these parasites might be having an effect on predation rates for these shrimp. That means we're even further underrepresenting what we think that, or what these food webs could possibly look like if we aren't counting in the parasites. And so I am all about thinking about parasites. And that's my bias. So there are just a few takeaways that I wanted you all to get from this. The first is that cylon affects shrimp susceptibility to epibiosis, and the reasons for that we're hoping to get to the bottom of. The second thing is that cylonized shrimp have shifted fatty acids that could alter nutrition. I didn't explain that too much, but if you have questions about that, you can ask me and hopefully I'll be able to give you an answer. But also our food web analyses and how we interpret fatty acid data. But these things really left us with more questions than answers, because we see an effect, but like a lot of the why and how questions are still there. But first is, of course, shrimp behavior affected by cylon, and hopefully we'll find that out with Sun's work soon. Second thing is, are cylonized shrimp actually more vulnerable to predation, right? All of the effects that I was suggesting were hypotheses, either through the sensing route or through hydrodynamics or both. And these are really excellent opportunities for experiments that I hope either I can follow up someday on, or if any of y'all are interested in following up, please do. I'm curious to find out. And then lastly is, how long do the effects of cylon persist? And this kind of gets to John's question about what happens after the scar. And well, I do know at least in one case that the shrimp can survive and live after that, but in other cases, the shrimp didn't do too well after they had been parasitized. And at least in that situation or that season that they were infected, they weren't reproducing. So who knows what kind of carryover effects that they might have as things go on. And I really hope people study the system more. So thank you. I have a lot of folks to thank that made this work possible, including collaborators, different folks that have supply assistance, the folks and facilities at OIMB and Friday Harbor Labs, especially during the time that I did the bulk of this work, which was last summer. I was really fortunate because folks at those institutions had put in a lot of work to make situations that were safe to do research. And so really this wouldn't have been possible, and a good chunk of my dissertation would not have been possible without those folks' efforts. So I'm really grateful. And then lastly, the funding sources. So if y'all have any questions, please send them over.
All right, thank you so much, Reyn. That was really great. We're getting a couple questions in, and I have a whole bunch of questions as well, but I'll try to restrain myself and let you go through the questions. So did you mention measuring age of shrimp?
Yeah, okay. So one tricky thing is that this was like a snapshot study of these shrimp, right? So I just, it was over a period of a couple of weeks that I was able to go out and collect shrimp. I knew I didn't repeat sample shrimp because I kept them all in the same place. But that meant that I couldn't really track age. And so in this case, I used kind of, as I think John is getting to, I use size as a proxy for age. And so one of the things that I did do was an analysis including both age and rhizocephalan infection, because I thought both of those could influence epibiosis, and it wasn't consistent amongst the different kinds of epibionts. And then as far as the infection of, or the rates of infection along with the size of the shrimp, I don't quite remember what I found with that, which makes me think that there wasn't a strong effect if I saw one. So apologies that I don't have a clear answer for that. But my expectation would be that the older shrimp is, there's a larger surface area to get hit basically by a parasite and more opportunities as it ages to get infected by a parasite. So it could go both ways.
So everybody online, continue to ask your questions. If you want to be unmuted, you can also let me know either by raising your hand or putting something in the chat, and I can unmute you so that you can ask that way. While we're waiting for other questions to come in, Reyn, I was curious about, you said that you weren't sure about survival yet. I was also curious about the option for them to be able to be reproductive after survival potentially, and whether we'd see a generational effect of infection. And I was just wondering if you wanted to hypothesize whether that's even possible for them to be reproductive after an infection.
Yeah, so I don't think it's been figured out. There actually haven't been very many studies on cylon generally. The general expectation is that if a shrimp clears infection after the externa has been released, that it can reproduce again. And in what it seems to be happening is that for shrimp that are reproductive, that are infected, it's more that I think hopefully I'm remembering this correctly, it's that the reproductive organs of the shrimp stop developing further instead of them actually totally degenerating. And so that's one of the reasons that folks call it sterilization as opposed to castration, is because castration would suggest that the shrimp's reproductive organs would be completely obliterated. And so I would suspect that in later reproductive seasons that a shrimp would still be able to reproduce if it's able to clear the rhizocephalan infection.
So kind of adding to that, John asked, do infected shrimp change sex? So
well, thank you, John. I have some evidence that suggests that these shrimp do become feminized through rhizocephalan infection. There's an odd thing with these shrimp and some other pandalid shrimp. Some folks might know that these shrimp are protandrous hermaphrodites. So they start off as juveniles, then develop into sexual reproductive males, and in later seasons develop into reproductive females. And the preliminary work that I have on that so far suggests that that the size that these shrimp, again, as a proxy for their age, gets shifted earlier when they start to develop female features when they're infected with the rhizocephalan. So there seems to be that effect, but it's really odd, especially because these develop into females anyways, whereas the crabs that we know about the feminization for are all gonochoristic. And so it'd be a really neat case for folks to get to the bottom of, trying to figure out how that feminization works in this context.
Nice. I think we skipped a question as well. Is it Taylor? Do you see that, Reyn?
Oh, Tyler's?
Tyler's, excuse me.
Okay.
Yeah.
Would a share mortality between shrimp/cylon be detrimental to cylon reproduction? Yes. So that is for sure a case, because it's I think folks have called it shared doom or shared demise. And so if you're a parasite, you really don't want your host to die in a lot of situations. And so it would be really maladaptive if the cylon increase predation rates. And so that's one of the things that I'm trying to think about, especially when it seems that through epibiosis, the cylon makes it so hard on the shrimp, how it's something that doesn't get selected out evolutionarily. It must be, I mean, it must be fine enough that the cylon can still reproduce, or that it can still persist in the population as a trait.
Yeah, that was one of my questions, was just kind of teasing out the difference between susceptibility because of movement, or susceptibility because maybe they're being targeted because of the diversity of their nutrition, or some way of those, like, is it a targeted predation event versus movement limitation?
Yeah,
predation.
that'd be so neat to find out. One thing that I'm really curious or hope that somebody could do, whether it's me or not, is to do a tethering experiment just to see if predation rates in the field change depending on whether these shrimp have parasites or not. And that would really answer some of those broader questions.
Kind of speaking to that, another question of mine is really early on in your talk, you had talked about having a different net to gather them to reduce biases, to make sure that you were not just capturing the slower, easier to capture shrimp. So you must have had an inkling that there was some behavior impact to infection. Can you speak to that a little?
Well, I had an inkling that there could be an effect, but mostly I was trying to cover my bases just in case. That was one reason that I used the net, is because I mean, for one, could see the shrimp as I was collecting them, but I also didn't want to use a trap because I didn't know if there was some change in their activity or their the way that they fed that would have changed how well they got attracted to a trap or fell in. And so really that net was the best way that I could think of to do it without having all of them skitter away and tail flip. So.
All right. I just wanted to thank you very much for sharing your work. It sounds like there's a lot more that we can do to learn about the system, and that you've done a lot of foundational work. So I thank you for that. For folks that are online, if you want to learn more, Reyn's contact information is on the screen now. So feel free, I'm assuming, to reach out if you have questions or thoughts or ideas for him as he continues his work. For everybody else that is online and listening, I hope to see you next week. Thank you so much for joining us. I really appreciate it. And again, Reyn, thank you so much for sharing your work with us. I appreciate it.
Oh, thank you all for having me. That's great.
All right, everyone. I'll see you next week, hopefully. Bye now.